Cancer Cell Biology, Part 1. In this last topic of this cell biology course, we are going to be focusing on cancer cell biology. And I first want to remind you of some statistics. So 1 in 5 humans in developed nations die of cancer. And this is quite a high number. But this is not why I'm focusing on cancer cell biology in this course. In fact, in the US, as discussed earlier, heart disease kills more people than cancer, although I believe that's about to change. I think it's also important to note here that on a global scale, cancer is probably much less important than you think. I know that we have all been touched by cancer. All of us probably have relatives, friends who have been touched by cancer in some way. But actually, on a global scale, for developing countries, for the third world countries, malnutrition and infectious diseases are much more important in that area. But the reason that I want to talk about cancer cell biology in this course is because cancer happens when cells break the exquisite rules that regulate their growth and cell division. So this should be intuitive to you. In young animals, cell division exceeds cell death and you have a net growth. In adult animals, cell division usually equals cell death. You have this steady state. You don't have this net growth. And this steady state comes about because there is this exquisite control on the cell cycle. We learned about that in an earlier topic in this course. And cancer is essentially a breakdown in the cell's ability to regulate its own division or its multiplication. So in the context of cancer, cells grow and divide in a completely unregulated fashion. And these cells that are doing these are sometimes referred to as tumor cells. They are sometimes referred to as transformed cells. They are sometimes referred to as neoplastic transformed cells or just neoplastic cells. So I will use all those terms interchangeably. So what I will be talking about is the cell that has changed in some way such that it can no longer regulate its own growth. So if a mass of these transformed cells remain localized, if they are perhaps surrounded by some kind of fibrous connective tissue, this kind of tumor is referred to as benign. And in general, they are not dangerous unless they become so bulky that they compromise the function of the organ in which they reside, or if they compromise the function of the organs nearby, or if they secrete some kind of biologically active substrate such as a hormone. And that's when these benign tumors become dangerous. But usually a complete cure can come about by surgical removal of these mass of the cell. Cancer is counted as dangerous and life-threatening when these transformed cells are classified as malignant or metastatic. What that means is that these cells have acquired certain changes that make them grow and divide even more rapidly and perhaps more than the cells that are found in a benign tumor, and they are not surrounded by some kind of fibrous tissue. So they are free to move and colonize other cells and other tissues and other organs, and usually they acquire the ability to become very motile. And in this case, a surgical removal of this mass of cells is usually accompanied by some kind of chemotherapy in order to try to kill these kind of cells. And we will talk a little bit about that at the end of this topic. So one very interesting way to look at the difference between the transformed cells or tumor cells and normal cell is to look at their behavior in culture. So at the beginning of this course, we talked about culturing cells when we were looking at methods in cell biology, and I mentioned that given the right environment, you could grow practically any cell in culture. You normally have to provide essential amino acids, vitamins, and growth factors, and In the discussion of the cell cycle, we also learned for stem cells, you need a surface of a feeder fibroblast or special cytokines. But given the right environment, you can grow practically any cell. Cells in general also require a solid surface to grow on because that mimics their interaction with each other and mimics their interaction with the extracellular matrix that surround the cell. This is sometimes referred to as anchorage dependence. So they must adhere to something or be anchored to something in order to grow. And cells in culture, the normal cells in culture normally display what is known as contact-mediated inhibition. What I mean by that, the cells will grow in the tissue culture dish as a single layer, and as they grow and fill that dish, they begin to touch each other and form junctions and adhesions. Once that happens, they stop growing. So you end up with a monolayer in the dish that's not growing anymore. So all of the properties that you see on the slide are of normal cell, not transformed cells. So let's take a look at what happens to the neoplastic cells. So in the top panel here is a scanning electron micrograph of a normal fibroblast, and you can see that they are very, very flat because they are adhering very tightly to the surface of the culture dish. And if you drew a cartoon of what they look like in a cross section, you would see a single layer of monolayer of cells. Now you can see here in the bottom panel a very dramatic difference in the transformed cells or tumor cells. You can see that they are much more rounded, and that's because they are not adhering very well to the surface and they don't exhibit this contact-mediated inhibition. So you can get these pile of cells. Sometimes they are called the focus of the cells or foci in the plural. So on this slide, and in the slide after this one, I have listed some of the changes that are commonly observed in tumor cells. And I group them into two categories. So the first category right here is the adherence abnormalities. So you have diminished adhesion of these surfaces, and that's why these cancer cells or transformed cells are very rounded, because they are not adhering very tightly to whatever surface they are growing on. And they don't exhibit contact-mediated inhibition. So once they touch each other, their growth does not stop. They just start growing and piling on top of each another. If you were to stain a cancer cell with phalloidin, which you will remember from our method section, is an actin stain, you will notice that the actin is very disorganized. Normally, fibroblastic type cells will have these stress fibers, which are long polymers of actin that are on the bottom, right near where the cell is adhering to the surface. You will see a reduced external coat of fibronectin, which is one of the extracellular matrix protein. And there is a very high production of what's known as plasminogen activator, which causes an increase in extracellular proteolysis. So both C and the D point to this one is the property of the cancer cells, and they don't really require adhesion to anything. Normal cells need to adhere to the surface of the culture dish or need to adhere to some kind of extracellular matrix protein, but cancer cells don't seem to do so. These are the adherence abnormalities. Then you have the obvious growth and cell division abnormalities. They grow to unusually high cell density. They don't actually require growth factors to divide. You have this loss of anchorage dependence. You have this loss of contact-mediated inhibition. The cells are sometimes referred to as immortal. In the beginning of the semester, we talked about growing primary cells in the culture, and I mentioned that there is usually a finite number of doubling of these cells, 50 to 100 of these doublings of normal cell. But cancer cells seem to be immortal, so they can continue to proliferate indefinitely. If you took these tumor cells that are growing in culture and you inject them into a susceptible mouse, for example, an immunocompromised mouse, they actually form tumors. So these are just some of the changes that you can see in tumor cells, but they are by all means not all of the changes that you can observe. So exactly what kind of changes are we talking about? In tumor cells, you actually have mutations in the DNA that gives the transformed phenotype or that contribute to the transformed phenotype. And one experiment that shows that is illustrated on this slide. If you isolate a tumor and then from that tumor you purify the DNA and transfected some normal mouse fibroblast and then allow them to grow in culture, what you will notice is that some of those fibroblasts will become transformed. In other words, they will start to exhibit a neoplastic phenotype. These uncontrolled growth, the formation of these piles of cells known as foci, are due to a lack of contact mediated inhibition. And if you take one of these foci and you inject it to some kind of compromised skid mouse as shown in the picture, you will get the formation of tumors. The SCID stands for severe combined immunodeficient mouse. They don't have any T cells or B cells due to severe mutations. The reason you need a SCID mouse to this experiment is because most of the time your immune system will detect and kill the cancer cells, and chances of getting a tumor is far less in a normal mouse. So in this very basic experiment that shows you that it is something in the DNA that confers the neoplastic phenotype to these transformed cells. The fact that alteration in DNA or mutations in these genes produced transformed cells was first described by Michael Bishop and Harold Vermus in 1977. They describe how a mutated form of a cellular oncogene, CSERC, carried by a tumor-causing retrovirus as a VSERC or viral SERC cause tumors. This monumental discovery honored Bishop and Vermus with a Nobel Prize for this finding in 1989. And that really illustrates the importance of this finding because most people have to wait decades for a Nobel Prize, and they only waited 12 years. Another example of a Nobel laureate who didn't wait too long for his Nobel Prize was Kary Mullis, who invented PCR in 1985 and won the Nobel Prize in 1993. So the time that elapses between your report of a discovery and the awarding of the Nobel Prize really indicates the importance of your finding. So anyway, there are two main classes of genes that contribute to the transformed phenotype, and they are oncogenes and tumor suppressor genes. So proto-oncogenes are genes that play a role in regulating cell growth, and they are positive regulators of cell growth. What I mean by that is that the job of their gene product is to drive the cell through the cell cycle from G1 to S to G2 through M and so on. So the proto-oncogenes are the positive regulators of the cell cycle. They drive the cell cycle. Okay, so proto-oncogenes are not mutated, but when they are mutated, now they are called the oncogenes. Proto-oncogenes are the unmuted version of the oncogene, and it's the muted version that we will see in a moment which contributes to the transformed phenotype. So what kind of mutations in proto-oncogenes are we looking at? We are looking at gain of function mutations. So what I mean by that is if you have a gene that positively regulates growth and it acquires a mutation that makes it better at driving the cell cycle. So you have a gain of function. That's what makes it an oncogene. And these mutations are sometimes referred to as dominant because you only need to have one mutation in one gene of the diploid pair. Remember, mammals or humans are diploid. You will need to have a mutation in one gene, one of the pair, to contribute to the transformed phenotype. The other major class of gene is the tumor suppressor genes, and their normal function is as negative regulator of the cell growth. So they inhibit cell growth. And we learned about that in the cell cycle lectures that there is a delicate balance between the positive regulators and the negative regulators of cell growth. And that's where you get this exquisite control of the cell cycle. So what kind of mutations are we talking about in tumor suppressor genes? In this case, we are talking about loss of function mutations. So if you have a gene that negatively regulates cell growth and you lose its function, then it can no longer inhibit cell growth. And then cell growth is promoted in that case. So this should be intuitive to you. And in this case, these mutations are usually referred to as recessive because you usually have to lose both copies of the tumor suppressor gene. You usually have to lose the function in both copies of these genes in order for it to have any effect on the cell cycle. So the nomenclature is kind of unfortunate. When I call these genes oncogenes or proto-oncogenes or tumor suppressor genes, it sounds like the function of these genes is just specific for cancer, but that's not the case. I have to stress that these are genes that have normal function in the cell. When they are mutated, in the case of proto-oncogene, they will have a gain of function mutations. In the case of tumor suppressor genes, you have a loss of function mutation when they're mutated, which contributes to the transformed phenotype. But these are not extra genes that we have that just cause cancer or don't cause cancer or suppress cancer. These are genes that are functioning in us every day. It's when they're mutated there is a problem. So they are normal genes and the nomenclature is maybe a little bit misleading. So now let me show you some numbers that I think that you will find interesting. In the lifetime of the human, there will be about 10 to the power 16 cell divisions. And the mutation rate, normal mutation rate, is about 10 to the power negative 6 mutations per gene per cell division. And this is in the absence of any kind of pollutant or carcinogen or whatnot. The mutation rate is dictated by the limits of the error rate of the DNA replication machinery. Okay, so these numbers here are in the absence of the carcinogens or mutagens in the environment. So if you do the math, if you multiply this out, you will find that every single gene that you have will experience about 10 to the power 10 mutations in your lifetime. And again, this number is if you are in a pristine environment without carcinogens or mutagens, and we know that's not the case. This number is probably much worse than it appears from this point of view. So the question is, why not does cancer happen so frequently, but why doesn't it happen even more frequently, right? 1 in 5 is pretty good when you are looking at these kinds of odds. Well, thankfully, and as it turns out, you require more than one mutation to disrupt the cell cycle. And in fact, it's been postulated that perhaps you need as little as 3 and perhaps as many as 7 independent genetic events, incident, mutations, or accidents in the same cell in order for that cell to be transformed from a normal cell to a neoplastic cell. So there is some interesting data that supports the hypothesis that you will need more than one mutations. One bit of data is epidemiological. And you can see here, if only one mutation was required to transform a cell to make it neoplastic, then there would have been no correlation with age, right? But in fact, there is a very strong correlation of cancer and age as shown here. So you can see the incidence per 100,000 individual. You can see that incidence rate up to the mid to late 30s and early 40s is pretty low. And then after you enter your 40s, you get this exponential rise in the incidence per 100,000. And this really shows that you need to accumulate multiple mutations in the same cell in order to have normal cell become transformed cell. And these data here also tells us why cancer is not so important in developing nations. It is because individuals in developing nations don't live long enough for cancer to be a problem. Again, malnutrition and infectious disease is much more important problem globally. So here is another laboratory experiment that supports the idea that the multiple mutations are required to cause cancer, and that is often known as synergistic effect of the oncogenes. So the MYC and the RAS are proto-oncogenes. So MYC, we did not talk about MYC, it is a transcription factor, and RAS we did talk about. Remember, RAS is an important intracellular signaling molecule that becomes activated through the action of receptor tyrosine kinases. So you should remember this from your cell signaling topic. This experiment was carried out in mouse where the MYC and the RAS proto-oncogene mutations were introduced in mouse mammary tissue by transgenic technique. These specific mutations transformed the MYC and the RAS proto-oncogenes to tumor or cancer-causing oncogenes. These MYC and RAS oncogenes were placed under control of a promoter from a mouse mammary tumor virus, also called MMTV, and the promoter could be activated by the hormonal stimulation, which will overexpress these oncogenes in the mouse mammary tissue. So when the scientists monitored these mice over time and calculated the percentage of tumor-free mice, so you can see that mice that are expressing just MYC oncogene remain tumor-free for quite some time, maybe under 125 days or so. And then you start to see tumors accumulating in these mice. So MYC is kind of a weak oncogene. RAS has a little bit more oncogenic potential. If mice are expressing just the RAS oncogene, you can see maybe first 40-45 days they are tumor-free, and then you start to see tumors appearing in these mice. And that's exemplified by the fact that the percentage of tumor-free mice are declining. But if you express both the oncogenes in the mice in the cell, you can see that there is a very rapid appearance of tumor or a rapid decline in tumor-free mice. By 150 days, 100% of the mice have tumors. And so this tells you that you need multiple mutations in order to really get the transformed phenotype. And that's actually good news. So here are different classes of proto-oncogenes and tumor suppressor genes, and they can be classified actually into 7 categories, and they are all here in this slide. So the first 4 are proto-oncogenes. A gain of function in those contributes to the neoplastic phenotype. And the last 3 are considered tumor suppressor genes because if you get a loss of function, that contributes to the transformed phenotype. So the class 1 are these signaling molecules, and that's pretty rare, and we are not going to consider these any further. But you can imagine that if a signaling molecule is mutated, that it may actually become a more powerful inducer of cell signaling. Then there are cell surface receptors, class 2. An example for this is receptor tyrosine kinase. Class 3 are the intracellular transducers, and we saw lots of these. RAS, RAF, MEK kinase, MAP kinase are examples of intracellular transducers that we have already talked about. And also considered as class 2 are the intracellular receptors, and these are the receptors for the hydrophobic signaling molecule, unlike that these are for the hydrophilic signaling molecule. And then you have the transcription factor, which is grouped as class 4. So those are the proto-oncogenes. So the class 5 are the cell cycle control proteins, and these are negative regulators of the cell cycle. Then you have the class 6, that is DNA repair proteins. Now, DNA repair proteins are not directly related to the cell cycle, but you can imagine that if you lose the ability to repair your DNA, so loss of function, that you will actually acquire more mutations. I think that should make sense to you. And that increases the chances of acquiring neoplastic phenotype. So they are grouped with tumor suppressor genes, but they are not classic tumor suppressor genes in that they are directly not regulating the cell cycle. The tumor suppressor genes include apoptotic protein as well. They are shown in as class 7. These are proteins that regulate apoptosis. We didn't talk about apoptosis in this class, but apoptosis is programmed cell death. So these are proteins that promote apoptosis or promote cell death. So you can imagine if you lose their function, then you can have a cell that grows indefinitely. So that summarizes the types of genes involved in cancer, and we'll talk about them individually in the next 2 sections. So this ends the part 1 of the cancer cell biology.